Good morning, students. Once you have finished with the Mendelism, we have to go further with the new discoveries in genetics, right? So what Mendel explained, just remind you once, then later we will start with the next part. What is that he explained is one character is controlled by a pair of alleles, that means one unit factor. We call it a gene. Means one character of every expression, maybe it is a color of the flower, stem and all, it is one character, it has one factor which is controlling its expression, means genotype is controlling its phenotype, right? This is what he explained first. But now what happened is, this is not true in all organisms. Like in humans you can see there are no characters which are followed exactly, many of them are not followed by according to Mendelism, right? Like any character you consider, only I told you one or two characters in humans, that is about eye color in Western people and hexadactylity, having 6 fingers, and tongue rolling. These are the characters which are following Mendelism, only dominance and recessiveness. But as you try to apply this character in other characters, this Mendelism to other characters, it is not happening. Why is it not happening? That is a major question, right? And one more thing I would like to really tell you, that you people are doing work nicely and many of you are asking me questions, many of you are asking me doubts also. This is a very nice thing, and whatever homework I give, you are doing it, but till now whenever I explain you about the Mendelian inheritance, I expect you to just take the pick whatever you have written and send me, right? It is just not to just check you, just to know that how far you are going right. First initially you write looking at the problems, later you start doing on your own. Once dihybrid cross or monohybrid cross or incomplete dominance, test cross, and yesterday I gave you one question about dihybrid test cross ratio, right? This was the pending thing yesterday I had left. Let you solve, and once you check this and find out whether you have really done it correctly or not. So what was the thing left was the dihybrid test cross, which I did not do and I asked you to do it. Dihybrid test cross. So already one basic type of test cross you have learnt in monohybrid cross. What is test cross then? It is the cross between F1 hybrids and the recessive parent. This is the first type of cross you have learned in monohybrid cross. Now the same thing we applied for the dihybrid cross also. What is that saying? You are crossing F1 dihybrid or F1 hybrid. Dihybrid. Okay? Dihybrid. Dihybrid, and you are crossing it with the double recessive parent because it is dihybrid. So it is a double recessive parent. You are crossing it, right? One example you have already learned in the dihybrid cross about the seed character. Seed character, right? Seed coat color. It is normally which is dominant? Seed coat and its shape, right? Round or the smooth one is dominant over wrinkled one, and the seed color, that is yellow, is dominant over the green color. Okay. So taking the double dominant character in P1, I am talking about first P1, I am not just started with the F1, right? Double dominant parentage, you are taking the smooth with the yellow seed color, first you are taking. Then you are crossing with the recessive one in P1, I am talking about, that is a double recessive character. What is the double recessive character? It is wrinkled with green color. You have crossed, you got F1. How will be F1? F1 will be having, what is the character? Smooth or the round with the yellow color. It is a hybrid one, because hybrids I told you will be having both the dominant characters. So hybrid is smooth and the yellow character. Now that F1 you are going to cross with the double recessive parent in test cross. So what is that F1 phenotype I told you? Phenotype, it should be F1 dihybrid, it should be smooth and yellow seed. Cross with, you should write always explanation. In explanation you should say that let us consider the seed character in pea plants. You cannot directly write yellow and smooth. What is yellow smooth? You don't know. If you are not perfectly explaining it, you should at least write seed of the pea plant, right? Then crossed to the recessive parent, that is the one having smooth and the wrinkled seeds. This is the recessive one. Wrinkled and green color. Okay. Then what is the genotype of this? This is what I am writing is, this is the homework what I have given you, that I am solving. Right? All the things you have done at this was the homework left yesterday, and if you have solved it, you should have posted me, or else you have to solve right now. Right? So genotype is capital S, capital S, capital Y, capital Y. This is the seed character. This is dominant, double dominant, both of them. But as most of you write this one, but what I said is F1 dihybrid. Where you people go wrong is dihybrid you should take. What is a dihybrid? Smooth is fine, capital S, but there will be a small s because it is a hybrid character, and capital Y and small y. This is the recessive character. This is hybrid smooth yellow. The one what I told you, capital S, capital S, capital Y is the smooth yellow, but it is the homozygous or the pure variety, pure breeding variety. But as we are taking F1 dihybrid, it is like this. You must start with F1 only. Don't start with P1 in this whenever they ask you test cross. Now crossing with a double recessive, wrinkled and green, small s and small y. So always mention the S with the small tail. Write with the small tail. Otherwise this S and this S will get confused. What about the gametes? Gametic generation you can get here. There are 4 types of gametes. What is the reason? It is because of law of independent assortment. So capital S, capital Y one gamete, capital S, small y another gamete, right? Small s, capital Y one more gamete, small s, small y one more gamete. Okay? These are the 4 gametes produced by this only. What about this one? This produces the gamete small s and small y. Okay? While crossing, what I do is I take this gamete down and cross this. Right? Directly I will do the checkerboard. Okay? One side male gametes this way, male gametes this side, female gametes this side. Okay? Then here is the small s, small y. Cross this. 4 combinations you are getting. Capital S, small s, capital Y, small y. Who is this? This is smooth and yellow hybrid it is. Okay? Next one. Capital S, small y, small y. This is again smooth because capital S is there, but it is not yellow, it is green. Okay? Because it is having small y, small y. Next is small s, capital Y, small y. What is this combination? This is wrinkled, and this is yellow because capital Y is there, no? So it becomes yellow. Then last one is small s, small y. What is this? This is the wrinkled and green character. So this is how you have to write cross. You have to write the checkerboard. Many a times I have seen students doing mistake when they write 4 gametes here. There also they write 4 gametes. It's not necessary. Think these are all pure breeding varieties. They are homozygous. They produce one type of gametes only. Only when they are hybrids they undergo law of independent assortment. If they are pure breeding, there is no law of independent nothing. Just only one gamete, one type of gamete is formed. Therefore, only one gamete are written here. It is producing 1, 2, 3, 4 gametes. That is producing single type of gamete. Then you will get, what is the ratio now? 1 smooth and yellow. Okay. 1 is smooth and green. Another one is wrinkled and yellow. And last one is wrinkled and green. Therefore, the test cross, dihybrid test cross ratio, phenotypic and genotypic is 1:1:1:1. This you have to remember. This is the important question for 2 mark or sometimes 3 mark question, because this is an expected thing to do. Write the cross and explain. In the checkerboard you are going to write all these, the phenotypes and genotypes. Okay. This is what I expected, which I gave you as a homework. If you are done, fine. Otherwise, now you copy it down. Some more such questions are there in your neat book or whichever neat question bank you have, in that all such questions depending on Mendelism are given. Just take their question number, write them down, and then if you do have doubts, you please ask me. Okay, that is your homework again. Okay, now take it down fully. Right, while writing always show this S with a small tail, small y like this, capital Y, normal y. Okay, so that's done with the Mendelism. We have finished with monohybrid and dihybrid cross, and we talked about the back cross, we talked about the test cross, monohybrid test cross, dihybrid test cross. So everything we have finished with your Mendelian inheritance. That's the first unit we finished. Now we are going to talk next part, that is about, what is it? It is multiple alleles. See, I am going with the chapters which are connected, because sometimes books also they are given after this linkage, crossing over and all. You can understand linkage and crossing over when you talk about molecular biology, where you talk about the genes, chromosomes, all that. Right now I am talking about multiple alleles. So what are these actually? Alleles. See, as we were learning about Mendel's inheritance, Mendelian inheritance, we came to one conclusion that every character is controlled by one pair of alleles. That is a single gene or a unit factor with one pair of alleles. But as I told you in the beginning of this class, that almost all the characters in humans and higher organisms are not following direct Mendelism like a dominant character and a recessive character, because they all are governed by more than one allele. Okay? Means that is called as the multiple allele. Means a character's expression is controlled by more than one pair of allele, is called as multiple alleles. They are all called multiple alleles. So, lot of examples are there. Let me give some, not one example I am giving you, just I am giving you how to understand this. Imagine one gene is there for the color, for black color. Anywhere you take, it's not one animal I am talking. B for the black coloration, right? Then small b for normally the no color, no pigmentation. So this is considered to be the dominant one, and we call this as the wild character, first form character. This is the last total recessive character. What happens as the time passes, the allele keep on modifying through mutations. It undergo mutation, this will undergo mutation. So if this double B is for the diploid character, this B goes on modification, B1, B2, B3 and all. Just I am giving you for your example. Before and at the end it comes to small b without pigmentation. If this is the character, this is called the wild allele. Okay? All these which are formed now are called as the mutated alleles. And this is the recessive one, recessive allele. This way, BB, okay, if you take BB, or B1, B2, B3, these are the pair of alleles, B4, small b, the recessive character. Now the question is, among these all alleles, for one particular organism, how many will actually contribute? If one organism X is there, it will have its character either B1 or B1, B2, B3, B4, something like this. Means at a given locus, see how locus, let me explain you. Always we have two homologous chromosomes. I hope you know this. This is one chromosome, this is the another chromosome. One is the maternal chromosome, one is the paternal chromosome. Right? Alleles are the positions where these genes are located. This may be called as a locus of this allele. This may be the height character or the color character. If it is, this is called as a loci or locus. Okay? At this position only the gene will be located. So in mutated alleles or multiple alleles, at this locus, it may be B and B1, it may be B2, B3, B and B3 or anything like that. Means at the given loci, only two alleles can be located. That is called the characteristic feature of multiple alleles. But all these alleles code for one character only. that is why we call all of them as a multiple alleles. More than one pair of alleles which are going to inherit or they are going to code for a single character. It is called multiple allele. This kind of inheritance is called multiple allelism. Many alleles are going to code for the same character. That is called multiple allelism. Okay. Now, here I said so many alleles are there coding for the character. So all these are mutated alleles. How are they mutated? They are mutated by the wild allele. Wild allele get mutated to form so many mutated alleles. So, but at given person, given character, only two alleles can be located. Means although there are many alleles, they all will not code for the same character. At a given loci, only a pair of alleles will be located. So let me list the characters of multiple alleles. So what are the characters? Whenever a character is controlled or it is expressed by more than one pair of allele, it is called as a multiple allele. These are called multiple alleles. Then what are this kind of inheritance is called multiple allelism. Then how multiple allelism will express or what are other characters of multiple alleles? More than one pair of alleles control a character. Fine. Always multiple alleles originate by the mutation of the wild allele. Means first allele will be getting mutated and forming so many mutated alleles. Next is at any condition at a given time only a pair of alleles can be located on homologous chromosomes. Right? And one more thing, in any given combination, wild allele is always dominant over the mutated alleles. Means this is always dominant over all these mutated alleles. So that is a characteristic feature of multiple alleles. Okay, now if you have not understood also totally, I will have to give you an example. Then only you will understand well. This is the just general explanation I give you. Now I will give you some of the example, one of the important example of human beings that are showing multiple allelism. What are the characters? What is the example? So this you are little familiar already about the example what I am going to give you. Under multiple allelism, okay, what is the character I will give you is the blood groups in human beings. So inheritance of blood groups in humans follow multiple allelism. So before entering into the multiple allele blood groups as the example for multiple alleles, that is blood group as the example for multiple alleles, we have to know first blood groups. Little background I'll give you. This you have studied in first PhD also actually. But still I would like to give you example or explanation about the blood groups. So blood groups, this is what I am explaining is the introduction for blood groups which you have studied in first year PUC. But still with that background you can continue now. Therefore I would like to give you the background of blood groups. Right? The blood groups were discovered by Carl Landsteiner. That's the name of the scientist. Okay? In 1900. He was studying why exactly blood, although they look alike, they can't donate one person to another person the blood. Right? Then what he did was he collected some samples from different people and he separated the plasma from the cells, and the plasma and cells he mixed with the different people. Right? And the two different types of plasma, he calls it as plasma as a serum. Right? But he found that the serum contains something called the antibodies in it. Right? Therefore, suppose the serum or the blood plasma contain the type of antigen, one type, it will have opposite type of antibodies. Therefore, he discovered certain 4 types of blood groups. Let me remind you, this was there already. Okay. First, some blood groups are given. What are the blood groups? Imagine a person's blood group is A. What will be its character? The antigens present in the plasma will be of A type. A antigen will be there in the plasma. Means in the blood liquid plasma, you will get antigen of A type. But in the plasma, that is a antibody will be already there in his plasma, that is anti-B antibodies. Imagine this is the blood, okay, blood smear we have taken. These are all antigens of A type. They are all there. Antigens are nothing but protein. Again and again we will start and use the word antigen antibodies in future, because you are going to learn immunity in the next chapter. So antigens are nothing but proteins, right? These are the blood proteins. If the plasma contain antigens of A type, then in their blood plasma you will have antibodies against B. Antibodies against B. So antibodies are also proteins that engulf the foreign antigens and destroy this. This is the basic knowledge everybody should have. So antibodies are the type of proteins that engulf the foreign antigens and they engulf it and destroy them. That's a basic thing. So if a person has antigen A and antibody against B in his plasma, his blood group is A. Okay? That's the first thing you have to understand. Next, if the person's blood group, I hope you all have knowing your blood, you are knowing your blood groups, right? So you may have A, B, O or AB blood groups, right? So if the person's blood group is B, then his antigen in RBC, I write in RBC, right? This is the RBC, in the RBC, this is the plasma, right? Antibodies in plasma I am talking about. So antigens on RBC or on RBC are of B type, and in the plasma, that's a fluid, you contain antibodies against A. Means, another example, if this is a blood, this is A blood group person. If it is B blood group, then on his RBC the antigens are of B type, okay, and in his plasma they contain anti-A. So these are the antibodies, okay. So that such person's blood group is B. So how do you decide blood group? Based on the type of antigen you have on RBC will decide your blood group. If you have A antigen, your blood group is A. If you have B antigen, your blood group is B. Then other thing what you have to learn is their opposite type of antibodies are there in the plasma, the liquid plasma. Now, some people do not have any antigens, neither A nor B. Means another type of blood group I'll give you. Imagine that somebody's RBCs are there without any antigens. Okay, there are nil antigens. Okay, let me clearly separate them. No antigens, then we call it as a zero antigen blood group. It is not O. O is just to understand it, we say O blood group. But O refers to the blood group whose RBCs contain no antigens. There are no antigens, so it is zero antigen blood group O. But what is special thing in these people, always antibodies against A, antibodies against B. Both are there. Therefore, here you have anti-A antibodies and anti-B antibodies also. Correct? So, O blood group people contain no antigens on their RBC, but they contain antibodies against A, antibodies against B in their plasma. So, O blood group people contain no antigens. Keep it in mind. Then, some people contain both antigens, A and B. So, A also is there, B also is there. Then their blood group is AB. Right? But what they don't have is, there are no antibodies in their plasma. There is nil antibody in their plasma. Okay? Keep it in mind. So, every blood group's importance is based on what antigen and what antibodies they have. So, I hope you understood now. 4 types of blood groups. This is in plasma. A, B, O and AB blood groups. 4 blood groups. A, B, AB and O blood group. Now, what blood group person can donate the blood to which blood group? Means, suppose you have A blood group, to whom you can give the blood during blood transfusion, or from whom you can receive the blood. That is story, right? Whether you can receive from B or not, why not, all these things you have to learn. Okay, as I told you, these are the way you have to decide the blood groups. Based on what is the antigen on RBC, what is the antibody in plasma. Right? If the antigen is A, then blood group will be A. antibodies are against B in the plasma. If it is B antigen, then blood group is B, antibodies against A are found, and if it is no antigen, then it is 0 blood group or 0 antigen blood group, that is O, and it has antibodies against A as well as antibodies against B. Then if it is AB blood group, the antigens will be against of A and B both type, there are no antibodies. Now the question, as I told you, you have to learn which blood group person can give the blood to which other blood group. Means what is the pattern of blood transfusion, means which blood group. Suppose one person is A blood group, whether he can give it to B or AB or whatever it is, that is called the blood transfusion. That is, the compatible blood transfusions has to be done, right? Actually before transfusing the blood, it has to be checked, right? Suppose one person has undergone severe bleeding, then if he requires blood, then somebody should donate him. Before donating, the blood group of the person who is injured, who requires blood group, his blood group has to be tested, along with who is the donor. Both the blood groups have to be matched, and then only it has to be transfused, right? So let us learn how it can be transfused. What is the way of transfusion? Means if you want to donate, what is the way they can transfuse? Which is the right compatible blood group to be transferred? Compatible means matching. Which is incompatible, which should not be transfused, right? That is why nowadays it's becoming compulsory to check your blood groups. Even you people have to check your blood groups, and because I observed most of you put in the DL, that is your driving license, sometimes you have kept the blood group thing blank. Don't do that. Always you should have the blood group in that. Check it properly and write your blood groups, right? Now, what is the donation? What kind of person donate to which kind of other blood group? So suppose the person is having A blood group, he can donate the blood to A because it is the same blood group, but he can also transfer to AB because AB person has no antibodies. Nil I have written. See here, nil. In AB there are no antibodies. Therefore, he can donate the blood to AB also. So person having A blood group can donate to A or AB. Both of them he can donate. Then while receiving time, if you want to write receiving person, if the person A person is receiving, he should receive only from A, and second person with whom he can receive is O blood group, because whenever O person give it to A, this person has no antigens. Therefore, this person will produce antibodies only against B. If O is transferred, there are no antigens recognized, then blood group is compatible transfer can happen. Therefore, O blood group person can donate to A also. Then if it is B blood group, he can donate the blood to B, and he can donate the blood even to AB because AB doesn't have any type of antibodies. Remember always this point. Then while receiving, he can receive it from B as well as he can receive it from O. So always whenever I write O, you have to think why O? Because O blood group person do not have any antigens. So B blood group person has antibodies against A. So if the O blood group is given, it is having no antigen, so it can be received. Therefore, B and O can be received by him. Then if it is O blood group, he can donate to whom? O blood group has no antigen. He can donate to A, he can donate to B, he can donate to O, he can donate to AB also. Therefore, he is a universal donor. A, B, AB and O he can donate. For all the blood groups he can donate. So if you are having O blood group, nice that you can donate to any type of blood groups, other blood groups. But don't think this is the only thing. After this I will explain you RH factor also. That also will matter. A positive, A negative, B positive, B negative. That whole thing you have to consider once. Then you should do the matching blood groups. You cannot donate A positive to A negative, A negative to A positive. There are so many constraints in that. Therefore, after learning blood groups, I will explain you RH factor also. Then only you can have the proper blood group, matching blood group to be transferred or to be received. Okay. Now, if it is O blood group, he can donate to all the blood groups A, B, AB and O. So we call him as a universal donor. He is a universal donor. This blood group is said to be universal donor blood group. This is asked for one more question. Which blood group is called as universal donor blood group and why? First thing, O blood group is said to be universal donor blood group. Why? Because O blood group person, in his blood there is no any type of antigen present on RBC. Therefore, he can donate to all other blood groups. But while receiving, he can receive the blood only from O blood group. Remember, only from O he can receive. Coming to the AB blood group, he can donate blood only to AB, nobody else. Because he has antigens both A and B. He cannot donate to either A or B. If he donates to A, there are antibodies against B. If he donates to B, he has antibodies against A. So he cannot donate to anybody other than AB. But while receiving, because he do not contain any antibodies, he can receive from A, receive from B, receive from AB and O. He is a universal receiver or he is a universal acceptor. Universal recipient we call it. Recipient blood group. So very important, AB blood group is a universal recipient, O blood group is a universal donor blood group. So therefore, if you have O blood group, you can donate, and if you have AB, you can only receive for any number of from all other blood groups. So this is very important. Before you get into the blood transfusion or transfer of blood, you have to check the blood groups first. Okay. So this is the first thing. Apart from this, I will give you now the genotypes of blood groups. Means what is responsible for the expression of blood group, right? That is what we learn under the inheritance of blood groups. So inheritance of blood groups. Now we talk about the term. What is the term? The term is about the genotype of the blood groups. And I told you blood group inheritance we call it. Okay. So that table, whatever I gave, you have to write the table properly and keep it with you all the time, because those questions will appear anytime either in board or in the NEET examination. Even in CET they have been asked. So coming to blood groups, we are talking now about its inheritance. How it is inherited. As I have started with this whole chapter saying that it is an example for multiple alleles. Means blood groups show multiple allelism. Now the question is, which blood group get what gene? Which gene expresses which blood group? That is what we call it as inheritance of blood groups. So there is a gene named I representing iso, hemo, agglutination. Whole thing is single word, I am just splitting it. Agglutination gene. This gene is responsible for the production of antigens on RBC, right? Then what are the various alleles of I, right? So that is what we learn under the heading genotype. If the genotype of blood group, if it has I, okay, if we write here I, with A as its superscript saying I gene has allele A. A is responsible for the production of antigen on RBC as A antigen. So if it is IA, it can produce antigen A, right? But the blood group will become, what is the blood group? Blood group will be A blood group. Now, as you know that there should be diploid condition. You should write a pair of alleles. Then if it is IA, it is homozygous for A antigen. That is a homozygous A blood group, right? But there is another possibility to have the A blood group, IA and IO. This is another possibility. Means if the genotype is IA and IO responsible for no antigen production. IA is for antigen A production. It produces antigen A. Therefore, it will become again A antigen only, because IO, although another allele is there, it has no any allele for production of any antibody. Therefore, this is a heterozygous blood group for A. This is a homozygous blood group for A. IA is a homozygous A blood group genotype. IA-IO is a heterozygous blood group for the A blood group. Okay. Now, second type. If you have IB, means the gene I has the allele B, then it will produce B antigen. What are the possible genotypes? IB is a homozygous B blood group genotype, right? Whereas IB-IO also is possible, which has heterozygosity, but it also will produce B antigen. This is again going to become B blood group only. So both of them result in B blood group. Next, okay, third one. If the blood group or genotype is IA on one allele, IB another allele, IA and IB if it is there, it is a heterozygous combination. Then these IA will produce A antigen, IB will produce B antigen. Therefore, it will produce both of the antigens A and B. Such blood group will be AB blood group, because here IA allele and IB allele both are codominant. This is called codominance. Very important word, you remember, codominance they show. This I told you when I explained you about Mendelism, deviation from Mendelism. I told you codominance. I told you there also that when I explain you about the blood groups, I will be explaining you about the codominance. So people having AB blood group show 50% alleles of IA type, 50% alleles of IB, and they will have both A and B antigens in their RBC. Therefore, the blood group AB is considered to be the codominant type of blood group. Both the alleles are present in it. But coming to the O blood group, it has no antigen, IO, or you can write small I, small I. So here also you can write small I instead of IO, right? Here you can write IA and small I also, IB and small I also. Both are same thing I am writing here. This refers to 0, means there is no allele for the antigen production. This will be no antigen or the nil antigens produced. Therefore, the blood group is O, right? O blood group is because of no allele on the gene I, resulting in no antigen production. Therefore, blood group is O. So this is the total genotype list of genotypes responsible for the formation of antigen and the blood groups, right? This table should be with you, right? Then you can later analyze how to address these, right? So how this is useful? One example I give you. Example 1. Okay? If suppose a person has blood group B, he marries to the blood group having O. What are the expected blood groups? They will ask, right? Here, person with B blood group has two genotypes possible. What are the genotypes possible? First one is IB, right? I write B and O here. Okay. IB is one blood group, one genotype IB, and IO has only one IO. Okay. Now gametes, when you write, it becomes IB, it becomes IO. Then combine them, we will get IB, IO, then they will have B blood group. Right? If the parents are with B and O blood group, and if the person having B blood group is homozygous for his alleles, then they can have the babies having only B blood group. How do you identify he is homozygous or heterozygous? Then you have to go for their parental genotypes or their parental blood groups. Suppose this is a man having B blood group, he has parents having B blood groups, both of them, then he will be homozygous. Right? You have to identify parental blood groups, means grandparent blood groups. Then only you will know whether he is homozygous or heterozygous. I told you that homozygous combination will get B blood groups. But if you write heterozygous combination, what you will get? Right? The same thing, just first you have to note it down. This one, this cross you should write it down. Okay, 1 minute I will give you, because this I will retain and write another one. This is very important table for you. Keep it always as your basic thing required, like how you learn logarithms, that way you have to learn this also, to learn the blood groups. Okay, I hope you have taken it down. Now I will give you another combination of genotype. If the B blood group person, if the genotype of the B blood group person is IB-IO, is heterozygous for B blood groups, then O blood group is common, IO, same. Now gamete formation, he will produce two types of gametes, IB 50%, IO 50%. Then here only one gamete because homozygosity. Now whenever you write the cross, what you have to do is, first one you take straight, second gamete you take it cross. This will be IB, IO. This is going to become B blood group. Fine? This is the first one. But whenever you write second one, you take this, write this as a cross one. IO, IO. This is resulting in O blood group. Okay? So now we got two opportunities to explain. One is, if the person having B blood group is homozygous for his B blood group, means he has IB, then O blood group is homozygous only, nothing else will be there. So the O blood group person will have IO, right? Then when the person homozygous for B blood group marries a O blood group, then the resultant children will be always with B blood group only. But if the parent is heterozygous for B, then it will be IB, IO. Then IO, IO, they can have two possibilities, B and O blood group possibilities. Right? Now one thing you remember, how to decide whether they are homozygous or heterozygous. If they don't give you the grandparental blood groups, then you have to think that any probable blood groups they will ask you, then you take heterozygous, because probability you have to do. Therefore, if you take the heterozygous combination, you will get all probable combinations. Now, how to analyze this? Most of the students will read this and write this way. Parents having B and O blood groups will get two children. One will be with B blood group, one will be with O blood group. This is wrong way of writing. This is not showing you two children. This is a probability of child having B blood group is 50%, probability of child having O blood group is 50%. Means whenever the mother is expecting the baby becoming B blood group, baby getting B blood group is 50% chance, and baby getting O blood group is 50% chance. This is how you have to analyze. Right? Most of the time I see students will look at this and write two children. No, it is probability. Therefore, the chance of getting B blood group is 50%, chance of getting A blood group is 50%. Therefore, it is 1:1 ratio. Means they may have either B or O blood group checked. Right? So now one more thing. Why the blood groups are required? Why you want to know? What is the significance of these blood groups? Very important thing. It will help to identify the biological parental genes or parents or paternity, mainly paternity. If you know the mother and the child blood group, you can guess the father blood group, right? And how it is useful, right? Everybody will know these are the parents, this is the child. There are some possibility where child is lost in childhood, right? Suppose it is again, it comes back and return to the parents. If it has grown up, then to identify the parents, right parent, father and mother and the child blood group is the basic test to be conducted, right? Then we can analyze. One more problem is, imagine, not now, old time when there were no good medical facilities available. That time, whenever in a maternity home there was a childbirth, normally if it is a midnight, around night, 12 o'clock, 1 o'clock, if the child is born, at a time, suppose imagine 2-3 children are born and hardly any doctors are there. At that time, there are chances that the born ones, young ones, newborn ones may get exchanged also. That time, parents will not even identify, they may take the wrong blood group child. Therefore, it has become mandatory now to check the blood group of the child and both the parents before they hand over the baby to the parents. And it is a compulsory thing, right? Because that kind of thing should be avoided, right? Therefore, paternity and maternity and child can be identified with the help of blood groups, right? Even in the place of crime, if there is a bloodshed and all, to check the blood group, it is a basic way to avoid innocent people to be caught as a criminals, right? Right person should be identified as a criminal and it should be punished. Blood groups are the basic things. Even I told you one more thing, whenever you are getting your license, that's driving license, rightly you put your blood group there. Before that you check your blood group in a small lab or I'll explain you how it can be checked also. We can do it on our labs also. That testing is very important, and put it on your ID cards or on your DLs, wherever their identity is required. That is one of the important identity of yours. Correct? So now, in this kind of combination, if the father is B, mother is O, children may be either B blood group or O blood group. Now, what is impossible blood group in this kind of, for these parents? If parents are with B and O, what are the blood groups which are not possible? Listen to me carefully. I am saying which blood groups are impossible in this combination. That's the thing you have to understand. So tell me now, you can only tell me, but now I will give you. In this, A is impossible, not possible is A, then AB is impossible. These are the impossible blood groups. Child, if at all, is A, that means it doesn't belong to these parents. That's a simple meaning. You can say whatever may be the reason. Maybe child is lost, whatever it is. Therefore, if the baby is A or if it is AB, that means it doesn't belong to the parents, simple meaning. Right? That's how it is helping to identify the paternity, maternity and or parentage we call it, and the children. Right? Whatever is not possible, based on that you can identify. Okay? So like this you have to take one or two examples and solve some more in this same pattern. Right? I expect this you to do. Right? And then get an analysis, this kind of possibilities for the children if the parents are like this. Some more I am going to solve now. Okay, keeping this as an example, one more I will give you now. What is the other possibility? Other examples I will give you now. One more quick example I will follow. Just you follow along with me. Then other things you can do it as a homework. Right? Second combination possible in this, it is, imagine parents. Maybe the father is having AB blood group. Okay? Mother is having O blood group. Find out the possible blood groups in the children. This is the example 2. Genetic problem number 2. First example I will already give. Second example I will give. Now, what are the probable genotypes? Only one genotype for AB blood group. It is IA, IB. What about O blood group? It is IO, IO or small I, small I. Now, what are the gametes? Here there is segregation. Two types of alleles are there. IA and IB. What about this? She will produce only one gamete, that is IO. Now, probability. This sperm may fertilize this egg. Right? Always take this thread, take this cross. Otherwise, you will write all crosses one above the other. It is highly confusing because I have seen some students will be writing one above the other 100 crosses. Then you will not write properly. Take this thread, take this cross. Right? Every step you follow in exam, it is very, very important, crucial to write proper crosses. IA, IO. What is the probable blood group here? It is A blood groups in children. What about this? You take this thread, you take this cross, it is IA, IB. What is the blood group? Blood group will be AB blood group. Right? Sorry, it is IB and IO. See, IB is here, this is IO. Observe, IB is here, IO is here. Here, IA is there, IO is there. Here, IB is there, IO is there. Right? Then, what will be the blood group? Possible blood group is only B blood group here. This is A blood group, this is B blood group. Now, observe one cute thing here. What is that? Parents are with AB and O blood group. But children don't have any parental blood groups only. Neither child is AB nor child is O. Right? The children are either A or B. This is the right type of child if the parents are AB and O blood group. understand. It is so curious to understand, right? Because I have observed immediately looking at this only that O is negative or it is recessive. Let me write AB. No, you should write this cross and get it. So if the parents are with AB and O blood group, parent AB is heterozygous, codominance, and this is IO, then this is segregation of alleles because of heterozygosity. Here one allele, first combination will produce A blood group, second combination will produce B blood group. Means the parents having AB and O blood group cannot have their blood groups in their children. Instead, they are having either A or B blood group. Therefore, looking at a person's blood group is A, you cannot say your parent must be A blood group. No. Looking at the child having B blood group, which cannot just suggest that parents are B blood group. This is also one possibility where the children may be either A or B blood group. They are both are heterozygous, but they are there, right? This way you follow certain examples and do some more examples like this, right? Let me check how we will do it. Okay, then some more examples I'll give you for you to do homework. What is that? The parental blood groups I'm giving you. Parents. What are the parents? First combination, you write A with A, homozygous combination and heterozygous combination. Okay, first combination, what is this, right? Second one, A and O, again A homozygous, heterozygous, you write. Then third one, A and AB, both homozygous, heterozygous. Fourth one, B and B, homozygous and heterozygous. Fifth one, B and O. Sixth one, B and AB. Seventh one, AB and AB. Eighth one, O and O, right? These combinations you try in all homozygous and heterozygous combinations. You do the crosses, each one cross. You cannot write directly here. You can keep this table and work out these problems. How to work out? So example 1, A cross A I have given you. A, what is that? IA and IA one combination. One combination you will get IA here and IA here. You will get cross IA. It is A blood group. First example is over. I told heterozygous combination. What is heterozygous? IO cross with IO. Then IA separates, IO separates. IA separates, IO separates. Right? Then write the checkerboard here. Whenever you get 4 gametes, you write the checkerboard. What is the checkerboard you are writing? One side you are writing IA, then you are writing IO, again you are writing IA, you are writing IO, right? IA is A blood group, then IO, you are getting A blood group, IO, again A blood group, IO, O blood group. So in heterozygous combination you are getting A and O blood group. Here you are getting only A blood group. This is how you have to fill this. Work out there and fill this. Work out there and fill this. This is your homework for the class. This is the most important thing. Once you solve it, you are done with this. You have won the whole thing. Right? This is a battle actually. Once you learn all this, then you have won the battle. Right? This I want everyone to work on this. Okay? I hope you will do this. All these combinations you have to work out, and after working out workouts and along with this, you make one small picture and send me. Right? Maybe it becomes 2-3 pages, no issues. Send one PDF and send me. Right? This is what I expect you to do. Once you do it, neat and all, so many questions appear on this, you are doing wonderfully well. Only thing you have to do the homework like this. That's why every day I am giving you one or the other homework. Apart from that, just writing the main examination, apart from that, some more things you have to do it to get into the main examination. Okay? So done then. In this you have finished now. We will now move on to the next part. That's about the RH factor. Just you are talking right now. about RH factor. Do you remember RH is the A positive, A negative, whatever we call now, that is RH factor, right? As you already know, the blood groups were identified by the scientist Karl Landsteiner. Now I am giving you RH factor. What is RH factor? Actually, this was discovered by Karl Landsteiner and Wiener. They, while working on the monkey named rhesus monkey, they were working on this. They identified, apart from the A, B antigens, there is another antigen called RH antigen based on RHE. This is the rhesus monkey. Based on this, they have taken the word RH. Therefore, RH factor. So, rhesus monkey, based on that they have worked and they gave the name RH factor. Right? What happens if you have RH factor in your blood along with your A and B antigens? Then if you contain it, then you are RH positive. Right? That is the antigen. RH antigen is present. You are all RH positive people. This is present. If there is a presence of RH antigen, then you are RH positive. If you have lack of RH antigen, then you are RH negative. So there are two blood groups. Either you may be RH positive if you have RH factor, you have RH negative if you don't have RH factor. Now, how many people have RH positive? In India, 97% people are RH positive. In Western countries, 87% people are RH positive. Right? This is Indian, this is a foreign thing. Then RH negative in India, how much? Only 3% left, remaining here you can calculate. Means very rare is RH negative blood group. Most of the students I have seen, they are saying I am RH negative, I am B negative, I am AB negative. There is nothing to be very precious about all this, feeling very happy about all this. Why it is problematic? They say it is very special. It is not special blood group, it is a rare blood group. The problem happens with them is they will not get the RH negative blood groups at all, because hardly 3% are RH negative. Therefore, they have to be more cautious. They should have the blood group list with them who are all having RH negative blood group. Now, what happens? Let me give you. Actually, if the RH positive person has antigen, that's all. He doesn't have antibodies in his plasma, right? If he donates the blood to RH negative, what happens? Right? If the person is RH negative, he receives the blood from RH positive, what happens? Actually, apparently, there is no reaction observed. Most of the time we will say, blood get clotted. No, it will not clot. Because then A and B, I told you, A and B, if you give the wrong blood group, then antibody will clump and agglutination happens, the blood will clot all over the blood vessels. Therefore, A and B and all you cannot donate like that. But what about RH factor? If you are positive, you give it the blood to the negative, there is no apparent reaction happening. Then what about the next one? If you give the blood from RH negative to positive, again my students always say that there will be clumping. Now I will tell you, there is no clumping here also. Because RH negative has no antigen. If it enters RH positive, there are no antibodies which are killing this, because this has no antigen. Therefore, no reaction. Positive person giving negative person, there is no reaction because this is negative, he has no antigens only and no antibody also. This person has antigen, but if negative person give it to the positive person, then because of negativity, no antigen here, he will not react. Now the problem is, if first time you have given the blood to RH negative person, in his body the antibodies start developing, right, which were not there but you provoked them. RH positive person is donating to RH negative person, initially nothing happens, but in his blood the antibodies start developing. Whenever second time this person donates the blood, then there will be blood clumping. In the blood vessels there will be clotting, and here and there clots are formed, blood vessels are blocked. Therefore, negative person, RH negative person should not take the blood transfusion from Rh positive for the second time. Even nowadays they will not give it for the first time also. This is a major important thing, major thing, because it's a life and death problem. It's not just like cool, okay, it's not cool. It's really difficult if at all any wrong blood transfusions are given. Therefore your basic work is not to have the blank place for blood groups in your identity cards or in your driving license. Wherever you have, you should have your blood group properly mentioned there, right? Now, why it reacts? Let me tell you, right? Now, what is the Rh problem? I told you right now. Normally, Rh factor is identified by the gene R, okay, capital R, okay. Small r is Rh negative. Those are Rh positive, they will have the gene R and R. Those who are Rh negative, they will have small r. These are the alleles, right? Actually, this also shows multiple allelism in the future because R1, R2 and all will be there. But this is the genotype, right? The problem when it happens is whenever Rh negative person receives the blood for the second time, right? Therefore, Rh negative person, if he receives the Rh positive blood transfusion for the second time, it is fatal. He may die, right? There is second problem happening. That's a significant. Second problem is something you have heard about this. I think its name is erythroblastosis fetalis. This is a kind of disorder. What happens in this? If the mother is Rh negative, okay, she is the woman having Rh negative, is the female, okay. If she married to Rh positive person, right, she is having RR, he is having capital R, resulting in capital R, small r, it will be R and R, the offspring will be Rh positive. I hope you understood, right? Rh negative is having a recessive character, positive is capital R, so you will get Rh positive fetus, okay. This is the mother, this is the father. If the mother has Rh negative blood group and father has Rh positive blood group, then the child will be, I'm sorry, it is the child become Rh positive. Very important now. The problem is, imagine mother is pregnant, okay, she has a placenta here and here is our fetus. This is umbilical cord. You know already umbilical cord you had drawn once. This is the fetus, okay. This is the fetus. This is the mother's womb, okay. This is a uterus. Let's imagine, okay. Here we have the outermost layer, okay. She is actually Rh negative. Placenta is having Rh negative. See, Rh negative placenta. This is Rh positive. All over here you have positive antigens. Baby is positive. During late pregnancy, whenever these antibodies, very important thing is, although chorionic villi are there, the antibodies produced may transport. Her negative antigens will pass and positive antigens from the baby will transfer. She will have some positive antigens in her whole body. She will develop some positive antigens. Yes, she will get it from the baby, right? Now she may start reacting. Placenta may start producing antibodies, but it is very slow reaction. Therefore, first baby is born, it will be normal. But once the baby is born, okay, lot of blood of the fetus will remain in the mother. So, lot of positive antigens will be there all over the mother's blood. Problem happens for the second child. If she becomes pregnant for the second child, baby is again Rh positive. Already she has some positive antigens. Then they start producing antibodies against this. These antibodies will pass all over here. They will break all the RBCs of the fetus. This is the second child. First child is born normal. Second child's all RBCs are broken down. Therefore, the name erythroblastosis. Erythroblastosis refers to erythroblasts are broken down due to the antigen-antibody reactions, due to which the baby will be dead. And the last dates of pregnancy. After 8th month also, it will have stillbirth, it will die. Or immediately after the childbirth, it may die. That is a problem. Therefore, successive all children may die because of this. Right? It is not, again, you have understood the seriousness of this, right? It's not so easy to just to have like this and all, it may die, It's not a just joke, right? This is the fact. Always science gives you facts. True facts are in science. That too, life science, it's a real practical. Now, what you have understood by this? Simple meaning. If the mother is a female or a girl is Rh negative, the best way to get married is Rh negative man or a boy, right? Otherwise, they may have problem with their children, right? It becomes incompatible marriage. So it is very important to check the blood group of the boy and girl before getting married. Whether you do it, all other jataka and all, you do it or not, their future you will decide or not by all those, the previous things what you are practicing. The most important thing you have to check is the blood group. If a girl is negative, she must get married to a negative person only. Rh negative is a good combination. Then children also will be Rh negative, right? It is negative, blood group will increase, right? But what are the other possibilities? Some students will ask, well, if the father is Rh negative, what happens? There is no issue. If the father is Rh negative, mother is Rh positive, then baby will be Rh positive naturally, because already you are seeing here. If the Rh positive baby is there, mother is Rh positive, same blood group, no issues. Therefore, it can carry on like this. It's not an issue. Are you getting this? Understood? This is the most important thing. I want you to work on this, right? This is the erythroblastosis fetalis, a dangerous disorder, and you will all have to study this properly. Now, one inheritance I'll give you. One example, another example on Rh factor, genetic problem. Example number 3 I am giving you. Identify the blood group produced in the children if the parents are with Rh positive blood groups, but if their grandparents were negative. Imagine a person having, or the male having Rh positive blood group married to the woman having Rh positive only. If their parents were having negative blood groups, then the genotypes will be, how will be the genotypes? They will be having R and R, small r. This is R and small r. They are heterozygous, right? Because there is a genotype variation. Then what is the gamete? There will be R here, small r here, crossed with capital R and small r. Then first combination, this combination, capital R, this will be Rh positive child. This combination, capital R, small r, this is again Rh positive person. These two combinations, capital R, small r, Rh positive baby. But last combination, R and R, it is Rh negative. So although parents are Rh positive, the child may be born with Rh negative factor. Understand this. This is slightly depth you have to do. If the parents, although they are Rh positive, they may be heterozygous for Rh positive. They may not be capital R, no. They are, maybe they are heterozygous. Then the children may be, possibility of child having Rh negative blood group is 25%. Having blood group of Rh positive, it is 75%. Therefore, this kind of blood group you have to work out, right? This is the most important thing for you to lead a healthy life, produce healthy children, right? Even you can suggest some people who are having Rh negative blood groups. And now one small simple way how to check the blood groups I'll give you now only, right? How to test the blood groups in the laboratory, right? I hope you have taken down this problem. If you have not taken down, you pause this and again take it down, right? Now the procedure to test the blood groups in lab. Okay, how you test is, there is something available in the market called anti-serum, right? This is anti-serum, refers to the blood plasma plus antibodies leaving the cells, artificial cells. This is called anti-serum. Now, they will ask you so many times anti-serum, meaning of anti-serum. Anti-serum is the plasma with antibodies. Now, such anti-serum are available in the market. You have to take them or you have to buy them. One is anti-serum A, second one is anti-serum B, third one is anti-serum D. Three types anti-serum I am giving you. Anti-serum D is for Rh factor. Okay. Now, blood group can be decided based on this. What all you need is, you need a sterile needle. The injection needle will be there, that one, or blood group testing needles are available, small ones, right? Then whose blood group had to be tested? That person's left finger. You take some disinfectant like ethanol or spirit with a cotton and clean the tip of the finger. Take the needle, just prick once, collect 1 drop of blood on 3 slides each, right? 3 slides you are going to take. First slide, second slide, third slide. One blood drop you are going to add, right? Now, anti-serum is available in small bottles. With help of ink pillar like things, add anti-serum A to this, anti-serum B to this, sidewise, not on the top of it. Anti-serum D to this. Now, take the toothpicks. Toothpicks are thin, stick-like things. Mix them thoroughly. Okay? Mix them thoroughly, and if while after mixing for some time, after you leave that for some time, if you find blood getting clumped like this, separating of cells and the plasma is happening, this is called as agglutination. If there is agglutination happening, that simply means this person's blood has antigen. See, it is anti-serum of A type, means it has antibodies against A antigen. So if clumping happens, that means A antigen is detected. His blood group may be A. Till you test B, you cannot decide. Suppose while testing, after testing B, there is no clumping. Blood is homogeneous liquid like, that means no agglutination, no clumping. That means he don't have any antigens of B type. That means only he has antigen A. So blood group is confirmed as A. Right? Now, in anti-serum D, that is for Rh factor, if there is clumping or agglutination, same like this with Rh, then you are A positive. If there is no agglutination, then it is A negative. I hope you understood. This is one. Same A, next one you take, again add anti-serum A. If there is no clumping, there is no agglutination, that means they don't have any A antigen. If there is agglutination happening, blood is separating, cells and plasma are separating, there is agglutination, that means B blood group is confirmed here. And in this again 2 types, if there is agglutination happening, this is B positive. If there is no agglutination, it is B negative. I hope you understood. Next, third one. If there is no agglutination, means he don't have A antigens. Again, suppose there is no agglutination in anti-serum B also, that means he has no antigens at all. That means his blood group is O. Then here again 2 possibilities. What are that? Agglutination with the anti-serum D, it is O positive. No agglutination with anti-serum D, it is O negative. Did you get it? Right? Understand thoroughly. Then suppose there is agglutination, like this separation is happening, agglutination is happening, that means A is there. Again suppose in this also there is agglutination, means clumping is happening, separation is happening, there is agglutination, that means he has both antigen A and antigen B, his blood group is AB, right? Again in these 2 combinations, if there is clumping, this is AB positive, if there is no clumping or agglutination, it is AB negative. See, everyone have to watch on this. right? I have observed some of the parents also looking at these videos. It is very important for every human being. You know that you are studying biology or not studying biology, it doesn't matter. But you need to learn all this, understand. Anyway, this kind of checking and all, we can do it only when you get the anti-serums from the market. This anti-serum A, B and D, all the three together, it comes in one pack, around 1500 rupees, something it is. Any scientific lab you will get it, but don't try it mostly in the house, because you may not maintain the needle sterility. Needle has to be properly sterilized. Then if you have it, you can try it. But what happens is, if you try it, you will get somewhat idea about it. At least you can try on your own, right? But anyway, it is little costlier. You can just check it in the labs also, or you can do some project when the college starts. We have one project on this. Those who want to check their blood group, they can bring their own needles, let them check in the lab. But anyway, this in the bracket it is. Don't think it is 100% going to happen. You can try on your own, but don't try first, at least learn, understand what is the pattern of agglutination. This is how our blood groups have to be analyzed, understood. So this is the important chapter I have finished on multiple alleles on blood groups, right? So this chapter you whole thoroughly you understand. Mendelism is over. Now multiple alleles is over. Next class I am going to start with the sex linked inheritance, sex determination, linkage and crossing over. That's a classical genetics will be over. Then we will go to molecular biology, right? So practice whatever homework I have done. I asked you, you do it and show me, right? That will be really easing your life. Okay? All the best, study well. Okay? Keep this in mantra in your mind. I am learning biology for my own life. Right? Then it becomes less risky if you have good knowledge about the subject. Therefore, live happily, think scientifically, develop scientific attitude. Don't keep on blindly believing everything whoever anybody is saying. Right? Therefore, be scientific, think scientific, have more scientific knowledge. Right? That makes you good, healthy human being. See you then.